Works. I'm going to get started right away. So I think this was the last slide we got to last Thursday, talking about enzymes. I'm actually just going to back up a little bit. And the fact, so enzymes, they're catalysts that increase the chance of any kind of reaction happening. The way they increase their reaction, the way they increase the possibility of that reaction happening is that they lower the activation energy. So the energy that goes in to start any type of chemical reaction, that's your activation energy. But if you have an enzyme present, you just don't need as much energy. So instead of like, my example was striking a match, instead of really struggling and putting lots of energy in to try to get that match started, that's a lot of energy that goes in, and maybe you just don't have enough energy to get it going. Where if you had an enzyme present, maybe you just needed to touch the match to that, matchbook, and it will go. And so you don't need as much energy to go in. It's more likely to happen. So a little bit more about enzymes, though. I think I got to the slide. So enzymes, they are proteins. They do have an active site. The active site is always where the substrate is going to bind. And my note, which is where we kind of ended up, is what substrate can fit into that enzyme is very specific. Most often, not 100%, but most of the time, the enzyme is named for the substrate that it fits with, which is my next example on here. It's just an example. You do not have to memorize any of this chemical equation. But enzymes, again, big blue or purple blobs, as in every book. This particular enzyme's name, and it's really long, you don't have to know it, it's called fructose 1,6-biphosphate aldolase. It's a mouthful. The majority of enzymes end in the word ase. So it's kind of like a big clue you're working with an enzyme. So it's called fructose 1,6-biphosphate aldolase because the substrate that fits in this particular enzyme is called fructose 1,6-biphosphate. So a lot of times they're just named for whatever substrate fits in. Once that substrate fits in to that active site, it's active, things are happening, it does form a more perfect fit. So it kind of went from a little bit wavy, curvy to a more exact, perfect fit. And this particular job of this enzyme is just to take that fructose molecule and divide it into two separate things. But my note, once that substrate is done dividing and breaking up that sugar into two molecules, because it goes from 1 to 2, nothing happened to the enzyme. The enzyme is right there ready to find another fructose molecule and break it down. And once it does, it goes and is all ready to find another fructose molecule to break it down. So a couple little things on my fill in the blanks. Enzymes are not changed. So they don't get changed whatsoever. So it's still the exact same enzyme. Or so once it finds its fructose, once it finds the sugar, it does its thing, but it's still right there to be able to do more of that thing. So they are recycled. Again, they're just there to keep doing whatever they need to be doing. They are specific for one substrate. There is not going to be any other molecule that can fit in that active site except that one very specific fructose molecule. They're specific for just one substrate. And they drive a reaction one way, meaning its job is to only break down fructose. It's not building anything. It's just doing one thing, one direction, breaking it down. And when a reaction does that, it's called a catalyst. It's getting its reaction from point A to point B. And that enzyme does it very, very well. There's some other things that can happen with enzymes. One, they can change how well they work. Again, enzymes' job is to get reactions happening by lowering that activation energy, but there are some things that change how well those enzymes do their job. One is temperature. Enzymes are proteins. Back to last class. Enzymes are proteins, and if you change the temperature on a protein, you might denature it. If you heat up a protein like you heat up an egg, it changes the shape of the protein and it can't do its job. And so if you heat something up, not only do you damage regular, structural proteins, you damage enzymes as well. pH, just having additional hydrogen ions or hydroxide ions can change how proteins are shaped as well. how much enzyme is present compared to how much substrate is present. If you have lots of substrate, very little bit of enzymes, the enzymes are going to do the best they can, but they can't break down every substrate. There's just too much of it. And enzymes are affected if there are anything called inhibitors around, which are my next couple slides. Because there's a couple different types of inhibitors that change how well the different enzymes work. This is showing the temperature. Enzymes work at best at certain temperatures. Most of the enzymes found in our bodies work best at body temperature. So too hot, too cold, they're going to stop working. Most of the enzymes in our body work best at a neutral pH. Again, that's what our bodies are. And then enzymes, as long as you keep giving them substrate, you keep giving them fructose, they're going to keep breaking down that fructose until you get to a point, it's called the saturation point, that it doesn't matter how much fructose you give that substrate. It's just, I'm full, I'm busy, you know, it doesn't matter how much more you give it. It's like it can't break any more down because it's so active already. Then your enzyme activity doesn't increase because it's maxed out. Of inhibitors that can affect how an enzyme works. One's called competitive, and the next slide, non-competitive. A competitive inhibitor, based on its name, it's competing for the active site. Again, active sites are very specific for what substrate can bind in there, but there's a couple sneaky little things that can try to sneak their way into that active site. So again, enzyme big blue blob. This enzyme's job is to fit that substrate into its active site, that big orange thing. But what if there's this little green shaped thing? Oh, it can fit in there too. And if this inhibitor gets there first, they're competing for it. If it gets there first, substrate can't bind. So it's almost like, you know, the race to the best parking spot. You know, whoever gets there first wins. Well, in this case, if the inhibitor gets there first, the substrate can't get broken down. It doesn't have a spot to, you know, fit into and get broken down. So competitive inhibition, just competing for the active site. Sometimes you have these molecules that can fit in there. Now, if it fits in there, no products are formed because the substrate can't get in there. It can't get broken down and make whatever products it's supposed to. Non-competitive, different textbook, different pictures. I like this one the best, but I think these are your textbooks. Different edition. Again, big blue blob. There's your enzyme. Its job is still to fit that substrate, that big orange substrate. But on one side of the enzyme, there is another spot that things can stick into. It's not the active site, but it's another spot things can attach to. It's called the allosteric site. You don't need to know that name. Just know that there's another spot things can stick to. Well, if these molecules, this little inhibitor sticks to that other spot, this enzyme changes shape. Just a little bit. Where that active site is meant for that substrate to fit in, that little active site, it sits there and just has that little flip over. So it looked like that, and it's just going to flip over just a little bit, just because this thing is sitting in there. So it makes the enzyme change its shape just enough that that substrate can't fit in there. It's not competing for the active site, so that's why it's called non-competitive. It's just because it doesn't need to compete for that active site space. It doesn't need to compete for that, you know, best parking spot. It just fits somewhere else. But because it changed its shape, the substrate can't bind there anymore because the active site changed its shape. And same outcome. If the substrate can't fit, can't get broken down, you're not going to get any product forming. Now, we do have something what's known as feedback inhibition, meaning there are times in our body we want enzymes to stop doing their job. It's like when we've made enough product. All right, we've broken enough fructose down. We've done enough of whatever it is. We don't need it to do any more. We don't need to make any more product. We've got enough. We can create an inhibition, and it's a non-competitive inhibition. So An example, if this enzyme, my enzyme 1's job is to take little orange circles and it will fit into the active site and they'll spit out little yellow triangles, and enzyme 2 is like, great, I love little yellow triangles, I will take them and I will turn them into little red squares, and enzyme 3 is like, awesome, I love little red squares, and I'm going to take it and I'm going to spit out little green ovals. You know, the end product is what our body needs to make. I mean, it uses multiple enzymes to eventually make whatever product it is. But at some point, like, we've got enough of these little green ovals in the body. We do not need to make any more. And so a lot of times the end product, when you get enough of it, becomes an inhibitor. This little orange oval will go up and attach to that first enzyme in the allosteric site, that little alternate site. So it's non-competitive, it's not competing for the active site. It will attach to that first enzyme, make that first enzyme change shape, and that first enzyme's like, oh, well, I can't take any more little orange circles anymore and turn them into yellow triangles. And if you don't have yellow triangles, you don't have red squares, and you don't have any more end product. So the idea is, once we've made enough end product, the end product becomes an inhibitor. Our body, you know, this is the most efficient way for our body to work. Makes what it needs to make, and then it stops. Now, it's the most efficient way. I like to call it the lazy way. It's like we only need to make exactly what we need to make, and then we're not going to make any more. Why go above and beyond? And this is one way our body can stop. Let's make it. Let's be done with it. That end product becomes the inhibitor. Now, most of the rest of this lecture is all on carbohydrate catabolism. Now, do you remember last week what's happening in catabolism? Do you remember my way of remembering it with the first 3 letters? Yeah, things are breaking down. So carbohydrate catabolism means we are breaking down carbohydrates. That's all it means. It's like a fancy way of saying we're breaking down carbs. Now, most organisms oxidize carbohydrates. Now, I talked last time about oxidation and reduction. Do you remember, and I have a little acronym up here, do you remember what's happening in oxidation? Something with electrons. One of them is giving, one of them is taking. But which one is this one doing? Electrons. So it's giving up electrons. And so when we break down catabolism, when we break down carbohydrates, we're also releasing electrons. We're losing electrons. And yes, other things are going to have to pick them up. Now, of all the carbohydrates that are out there, one of the most efficient ones that's broken down. And when I say efficient, it means we're going to get the most energy for that one molecule of sugar. It's like the most bang for our buck, and it's glucose. Now, there are 2 ways we can break down, catabolize, 2 ways we can break down glucose. One is through cellular respiration, and the other is through fermentation. We're going to focus today, we'll talk about fermentation, but we're going to focus today mostly on the cellular respiration. Now, in cellular respiration, there are aerobic processes as we break it down, and there are anaerobic processes as we break it down. What does aerobic mean? Is needed in part of that breakdown. Anaerobic, it means it can do it without any oxygen. Whole breakdown, but we're going to further break it down. So again, we can take glucose, which is going to be our very, very top. I wish this was even taller a little bit. I've gotten so used to my note holding it the opposite way because this has a magnet, and I would sit there and do this, and then it sticks. It bugs me. Side point. Glucose is my starting molecule. I can either take that glucose way at the top, and I can go into the green section of this picture, and I can break it down through cellular respiration. Or I can take that same glucose molecule, and I can go towards the orange section, and I can break it down through fermentation. There is a lot more steps in cellular respiration, and I'm going to draw a lot of stuff on my board. Compared to fermentation, there's just not as much going on. But respiration, the whole green part, goes through multiple steps as we break down the glucose. And our ultimate goal is get as much energy, ATP, out of this glucose molecule that we can. And so it's going to go through what's known as glycolysis, this little lighter green square. It's going to go through the Krebs cycle. and it's going to go through an electron transport chain. It's all the green part. Now, you have to know this? No. If you're like, I love learning about this, great. Take like cellular biology, which we don't even offer at Western. Take it at UWL. And you can spend the entire semester learning about cell and glycolysis and everything it does. What I want you guys to know is the basics. So we're really looking at what is going to be our starting molecule, what is going to be our ending product in this, just glycolysis in itself. What are some things we get out, and what are some things we put in the process? I do not care about all the intermediate steps that get us there. I want the beginning, the end. What are some things we get out? What are some things we get in? I'm going to draw some stuff on the board, and it's pretty much, I wish I could like raise that up a little bit, but I'm going to just draw near the bottom, pretty much go across most of the board. You guys should have a white sheet of paper, and I'll make sure you guys get it filled in with the squares on like what's going in, what's coming out, what we get out of it, that table. But what's nice, if you don't have a sheet of paper, you can use the back side of that as a blank sheet of paper to draw what I'm drawing on the board. And so in glucose, or carbohydrate catabolism, we're going to break down glucose. Now, just as they have on the very, very top up there, sorry if it's hard to see because it's bright in here, there are there are 6 black circles. They all have a C in them because they all represent a carbon molecule. Now, I feel like I asked this like a week or 2 ago. Does anyone remember what the formula for glucose is? It's C6H12O6. And again, our job is to break this down. And most of what we're going to focus on is where are all those carbons going as we break it down. That's where we focus. And so my 6 little black circles represent those 6 little black circles on the very top that have C's on them. These are my 6 carbons. And I'm going to break them down. So first by note, before I can start any chemical reaction, what do I need to put in? Enzymes lower it, but what do I need to put in? You need to put in energy. So at the very, very top of the diagram, we have a couple ATPs that are going in at the very top. How many ATPs are going in at the very top of that diagram? We'll get there. So at the right where it has that number 1 listed, there's an ATP going in, and right where that number 2 is listed, there's another ATP going in. There are 2 ATP going in. This is my little representation. 2 ATP are going in. Because again, we can't start anything off with the first putting in some activation energy. So we're going to put some get this going. And our end product, way at the bottom down here, are 2 molecules called pyruvic acid. And each pyruvic acid contains 3 carbons. I was like, I wonder if I can pull the one drape down, because I feel like it's really bright up there. So sunny though. Now, so we have our starting molecule, our glucose. We have our ending product of 2 pyruvic acid. Stuff out. All we've done so far by breaking down glucose, I don't know if that helped her. All we've done is put energy in, and our whole job in catabolism, when we break stuff down, it releases energy. How many ATP, looking up there, do we get out total? Like how many ATP do you see coming out? Total. So we have 2 ATP we gain right here, and we have 2 ATP that we gain right there. So using lots of colors today, we get 4 ATP out. What are some other things that we get out? Yeah, water. How many waters? Waters. They look like little upside down Mickey Mouses. We get a couple waters out. Ooh, that's not a great blue. I'm going to throw it out after this today. What else do we get out? One more thing. NADH, how many? 2. Why do I care about NADH? Do you remember from last class what NADH is? You might have to scroll back through your PowerPoint or notes. They're electron carriers, and we care about electron carriers today. So we things out of here, but we've also put some stuff in. So filling in your glycolysis column on that table. First, where does this happen? So where in the cell does glycolysis happen? And it happens in the cytoplasm. So it just happens in the fluid. It doesn't matter if it's a prokaryotic cell or a eukaryotic cell. Both cells have cytoplasm, so it happens same place. What goes in, well, there's 2 things that goes in. 1 glucose, I probably could write that up here. This molecule goes in. And 2 ATP, because we needed some activation energy. Longer list. 2 pyruvic acids, 2 water, ATP, and 2 NADHs. We've got 4 different things that come out. They are, what do I say? I mean, they're not half. An ADP is ready to pick up an extra electron and an extra phosphorus to become ATP. So we're kind of priming it. It's almost like we emptied the ATP of its energy, and it's just waiting to become ATP later on. And like, are the same ATP at the beginning? I think I'm confusing your question. So they'll become ADP. They can get turned into ATP like anywhere in the cell. It doesn't have to be just in this particular process. They're there and ready to pick up an ATP. They could do that right down here, but they could be that they're not attached to this particular formula. Does that make sense? I mean, they just be kind of like ADP floating out ready and primed to become ATP. For the energy produced box, so we made 4 ATP, but we also put 2 ATP in. So how many have we actually gained from glycolysis? 2. So we've got a net of 2 ATP, which is really not a lot. I mean, we get some ATP from glycolysis. It's just not a lot. For my anything else important, I kind of changed it. The biggest part is glycolysis will feed into cellular respiration or fermentation. So I'm going to go back to this picture. So glycolysis, this middle box, this glycolysis of taking glucose and making 2 pyruvic acid is either then going to feed into cellular respiration, but it also can feed into fermentation. So it doesn't matter which way, glycolysis is part of both cellular respiration and fermentation. It's that first step. So for that last, is that part of fermentation aerobic or anaerobic respiration? All 3. we're going to do glycolysis and go into fermentation. We can do glycolysis if there's oxygen around. We can do glycolysis if there's not oxygen around. It's just a quick, simple breaking it down. The word glycolysis, lysis, means to break apart, so it just means breaking apart glucose. It's just we split it into two, essentially. Now, that pyruvic acid that we made, again, it could either go into fermentation or it could go into the cellular respiration process. I boxed out fermentation because we're going to take it and we're going to go through the whole cellular respiration process with it. So we're going to take our 2-pyruvic acid and we're going to make something called acetyl-CoA. These are my next several slides. Then we're going to take acetyl-CoA and we're going to go into the Krebs cycle. Then from the Krebs cycle, we're going to go into the electron transport chain. That's our final series of giving up and picking up electrons. So, making of acetyl-CoA. This is also, see worksheet, where to fill it all in. So pyruvic acid, alternate name for it is pyruvate. Different textbooks. Don't care, just as long as you kind of recognize the name. Is taking pyruvate, which is a 3-carbon molecule, and turning it into something called acetyl-CoA. So I'm going to take this 3-carbon molecule, I'm going to get rid of my 2, we'll put 2 over here, and turn it into a 2-carbon molecule called acetyl coenzyme A, or just acetyl-CoA. First step of cellular respiration, once we've got that glycolysis down. Pyruvate, pyruvic acid, turned into acetyl coenzyme A. Now, there are 2 pyruvic acids here. I'm only going to follow one of them through because I really don't have a lot of board. Just know, whatever is happening to this pyruvic acid, remember it's happening to the other one too. I'm just not going to keep drawing 2 acetyl-CoAs and 2 Krebs cycles. So when we start adding things up on what do we get out, just know whatever I get out here, you're getting another one also out in, my imaginary, not drawing it on the board. So pyruvic acid turning into acetyl-CoA. So the first, we're just going to go, where does it happen? This also happens in the cytoplasm of the cell. It doesn't matter what kind of cell, all of cytoplasm. For the what goes in, 2 pyruvic acid. So the starting molecules, those 2 pyruvic acid is what goes in. What comes out? There's a few more things. One, we had 2 pyruvic acids. Each pyruvic acid is going to make an acetyl-CoA. So essentially, we're going to have 2 acetyl-CoAs coming out. What else do we get out based on the picture? Get CO2. I need another color. Go green. Now, this pyruvic acid, again, we go from a 3-carbon molecule to a 2-carbon molecule. That's where that carbon goes. I'm like, we didn't magically lose it. That 3rd carbon that we lost leaves as carbon dioxide. So this pyruvic acid gives us a carbon dioxide. The other pyruvic acid that I'm not drawing gives us another carbon dioxide. So how many carbon dioxides do we get? 2. This one, and then the other one I'm just not drawing. What else do we get out? ADH, and how many total? 2. The one from this one and the one from the other one I'm not drawing. And why again do I care about NADH? What's NADH? An electron carrier. So I've made NADH, which means these are now full of electrons. So what do I get out? I get 2 acetyl-CoAs, 2 carbon dioxides, and 2 NADHs. Do I get any ATP? No. So for the energy produced, none. We don't make any ATP at this point. That doesn't mean it's a worthless phase. One, I still needed to make acetyl-CoA, and I got some electron carriers that are just ready. This is also one of the reasons why we breathe out carbon dioxide. For the anything else important, The acetyl-CoAs are going to feed into the Krebs cycle. So once you make acetyl-CoA, they will immediately go into that next step. Is it part of fermentation, aerobic or anaerobic respiration? It is part of aerobic or anaerobic respiration. Now, and this is where some of the differences come from advanced A&P when you study this. Everything is usually like, no, it's all aerobically. That's because we're aerobic organisms. There are bacteria that can do this that are anaerobic organisms. And so if you're an anaerobic organism, you can do this anaerobically. It's not as efficient, but it's part of aerobic or anaerobic depending on the organism. Now, Krebs cycle. Do I expect you to know all of that? Nope. But some of the things I'm going to focus on and see where I can draw this. Really wish that thing was higher. One, if you notice at the very, very top of the Krebs cycle, there's your acetyl-CoA molecule. It's a 2-carbon molecule that's going to feed into the Krebs cycle. Now, as it feeds into the Krebs cycle, you have a 2-carbon molecule that's going to meet up with this molecule, which is 4 carbons. There's 4 little circles there called oxaloacetate. Don't care that you know the name. But you're going to have a 4-carbon molecule meeting up with a 2-carbon molecule. And when you have 2 plus 4, it equals 6. You're going to make a 6-carbon molecule. And that 6-carbon molecule, this is the only one to know the name of, it's called citric acid. Great. There's your citric acid. This is why sometimes the Krebs cycle is also called the citric acid cycle. Krebs because it's named after someone that discovered this whole cycle. Citric acid is because this is the big 6-carbon molecule that we just got done making. As soon as the acetyl-CoA goes in, mixes with that 4-carbon molecule, and we now have a 6-carbon molecule. Now, you know, lots of things are going to come out. This is my non-circle, my oval. This cycle is going to undergo lots of different changes and things like that, and eventually it will go right back to being that oxaloacetate. But I care, as that 6-carbon molecule goes back to its 4-carbon molecule, what are some things we get out? And there's multiple things. What's one thing we get out? ADH. And how many? If you look all around at that cycle, you got to find them all. We get 3, because there's one here, and one way up at the top. So we get 3 for each Krebs cycle, but how many Krebs cycles are taking place? 2. I'm just not drawing it. So like this pyruvic acid goes in there, but the other pyruvic acid would go into another Krebs cycle. So how many NADHs do I get out total? 6. 3 in this cycle and 3 in the other one I'm just not drawing because I don't have enough room. So we get out. Yep. Water's going in. Waters will go in. There's a little water molecule. There's one water there and another one in the other Krebs cycle. 2 waters go in. Waters are just helping break down the molecules. What else do we get out? Got 3 more things. What is it? FADH. How many? 2. We get this FADH2 out here, but then we got another one coming out in the other Krebs cycle that's just not drawn. So 2F. Why do I care about FADH2? What is it? Same as NADH. Yeah, it's an electron carrier. Yeah, because my next step is the electron transport chain. So I care about electrons. So I've got 2 FADH2s, I got 6 NADHs, all just from this one step. What else do I get out? More things. At the very bottom here, I'm going to get ATP. How many ATP total? 2. And then the one in the other Krebs cycle that I'm not drawing. One more thing that it's kind of tricky just based on how it's drawn. Not the COA, but it starts with a C. Some carbon dioxide that also comes out with the NADHs. How many carbon dioxides come out total? Like, or I should say, how about just this Krebs cycle? How many CO2s do you see coming out? 2. How many total? 4. Now, it is at this point, for the carbon dioxide wise, we started with the 6 carbon molecule. 2 of the carbons just lost there. The other 4 carbons, I lost here. So we've accounted all carbons from glucose, all get breathed out as CO2. So, counting up, what goes in, what comes out, and where does this take place? The Krebs cycle, for where it takes place, does make a difference. I write small in this little box. This takes place in the cytoplasm of a prokaryotic cell. It takes place in the matrix of the mitochondria in a eukaryotic cell. On A and P, you would have been like, nope, Krebs cycle takes place in the matrix of the mitochondria. Remember, prokaryotes don't have an advanced organelle like a mitochondria, so then it just takes place in the cytoplasm. For what goes in, ways are going to go in, like 1 in each Krebs cycle. And 2 waters are going to go in, which just help break things down. There's only 2 things going in the Krebs cycle, 2 waters, 2 acetyl-CoAs. Stuff out. We get 4 CO2s, 6 NADHs, 2 ATP, and 2 FADH2s. So 4 different things are coming out. Let's draw something, ignore me for right now. Dude, we make some ATP? Yeah? And I'm like, we made 2 ATP. Again, the Krebs cycle does not give us a lot of energy, but we made a couple ATPs. Ignore me, I'm just drawing. For the anything else important, the biggest, and this might actually be on my next slide. The important with the Krebs cycle are how many electron carriers we've gained. We only made 2 ATP, but we've harvested a lot of electrons into those electron carriers. So a lot of the energy that were in the bonds of that acetyl-CoA went on to making NADHs and FADH2s. We made lots of electron carriers, or I guess filled up the electron carriers. The other important part about the Krebs cycle is that a lot of these intermediate steps, these different acids, are used, they're building blocks that can be used elsewhere. So as we do break down different things, this succinic acid or the ketoglutaric acid, they're used as building blocks in other things. So they make precursor molecules for building other things up. Is a part of fermentation aerobic or anaerobic respiration? Aerobic and anaerobic, depends on the organism. Again, in A and P, you're going to be like, no, this happens only aerobically. Again, it's because we're aerobic. But bacteria that are anaerobic can do this anaerobically. Now the electron transport chain, the last step of cellular respiration. Where does this happen before I start drawing more stuff? This happens on the cell membrane of a prokaryotic cell. So this would happen on the outer membrane, because bacteria don't have a mitochondria. But they still need that phospholipid bilayer, which is found on that outer cell membrane. So I'm drawing phospholipids here. Of a prokaryotic cell, it's going to happen on the inner membrane of a mitochondria in a eukaryotic cell. With my little note, if I had to draw a mitochondria, they're usually orange in books. Mitochondria have an inner membrane. So if you're a eukaryotic cell and you have a mitochondria, it would be happening on that inner membrane, which is still phospholipid bilayer. Bacteria, outer membrane, because they don't have mitochondria. Now, the what goes in, it's called the electron transport chain for a reason. So in all of these beginning steps, we made a lot of electron carriers. All those electron carriers are going to head to the electron transport chain, so this is where you guys have to do math this morning. So one of our electron carriers was NADHs. How many total NADHs have I made from all the other previous steps? If you add them up. How many NADHs? 10, because we had 2, 2, and 6. So 10 NADHs go in. That's one of the in. So the electron transport chain, our 10 NADHs we've made previously are all heading straight to the electron carrier, or the electron transport chain. What was our other electron transport, our electron carrier that is going to head here? Yeah, the FADH2, and we just had the 2 of them. So the what goes in, 10 NADHs, 2 FADH2s. As you guys are going to notice throughout the semester, I am not an artist whatsoever. Because I never judge anyone else on their art. I'm going to come back to this slide for the what comes out once I explain it. It's phospholipids. So again, whether this is happening on the cell membrane, on the prokaryotic cell, or on the inner membrane of the mitochondria, all those electron carriers are going to head to that membrane. So here's that phospholipid bilayer. Here we've got proteins, enzymes, again, they're all big blue blobs. The idea is those FADH2s and NADHs, they carry electrons, but they also carry a hydrogen with it, which is why they always end in NADH or FADH2. So they carry the energy of the electrons, but they also carry those extra hydrogen ions. When an NADH or an FADH2 comes along, they use the energy in that electron to pump hydrogens across. So they use the energy in the electrons have energy, to pump lots of hydrogen ions across, like a ton. Again, you've got lots of NADHs, lots of H's, lots of FADH2s, again, H's. And so we use the energy in the electrons, we pump hydrogens to one side. What ends up happening is when you have a whole bunch of hydrogen ions on one side, they become quite crowded. Do they like to be crowded? Nope. So their job is they want to sneak back as easy and fast as the way they can. They would like to go to the other side where they are not as crowded. And so what they're going to do is they're going to sneak past through this particular enzyme. I was trying to draw that. This enzyme, and guess what name it is? ATP synthase. And most enzymes end in ase in their names. Based on the name of the enzyme, ATP synthase. When you synthesize something, it means you make something. So the name of this enzyme truly means making ATP. Now, when this hydrogen ion sneaks back through, top of this enzyme will spin like a turbine, just like a wind turbine spins. So when the hydrogen ion sneaks through, it spins the turbine. So that's what it's trying to show there. Now, just like a wind turbine spins, that's generating energy by spinning that turbine. So the hydrogen ion spins the turbine. It now has energy in it, and that energy on that spinning turbine is used to attach a phosphorus to ADP, and it makes ATP. Staying consistent with my colors. So it's this enzyme that's going to be turning because of what's known as the gradient difference. Gradient just means a lot on one side, little on the other side. And because there's a lot on one side, they want to sneak back. They want to spin that turbine, and that turbine makes ATP. Depending on how many hydrogen ions can sneak through, because some, they have leaky membranes and they might be able to sneak through the phospholipid bilayer. But most of the hydrogen ions are going to go through this ATP, or this ATP synthase enzyme. And as it spins, it churns out about 34 ATP for every glucose molecule. So all of our NADHs, all of our FADH2s create this gradient. As it travels through, it spins the turbine enough that that turbine makes approximately 34 ATP. And I say approximately because not every hydrogen ion goes through there. Some will sneak through else in other areas, and then they don't spin the turbine, and you may make less. But it makes on average 34 ATP. So for the energy produced, well, let's get for what comes out, because I want to make sure I get you filled in. What comes out? We get ATP. We also get water. It's because those hydrogen ions, we still have hydrogen ions, and as long as there's oxygen around, meaning it's an aerobic process, if they're doing this aerobically, we will make some water. We make ATP and water. Energy produced, 34 total ATP. Give or take a little bit. We go with the average. Are there anything else important? So the electrons. Again, we use the energy of the electrons to pump those hydrogen ions across. But those electrons and their energy do have to go somewhere. We don't like to have floating electrons around. They do have to find a final electron acceptor. And the final electron acceptor is usually oxygen. This is where we make water. It's not always. It gets into fermentation. But the job of the electron transport chain, pump hydrogen ions across the membrane. We pumped a whole bunch of them. And then the ATP synthase enzyme makes ATP, it's in its name, because of that hydrogen ion gradient, just meaning lots of hydrogen ions on one side where they're crowded and they don't like it. And they want to sneak back. Is this part of fermentation aerobic or anaerobic respiration? It is usually aerobically. Organisms that are anaerobic can do this. They will not make as much ATP. They will not be generating 34 from this step. But it can take place anaerobically, but it's usually aerobic. Now, if you add it all up, how many ATP do we get total out of breaking down one glucose molecule? Make 38. Again, on average. Some bacteria do this more efficiently, some not as much. But on average, we make a total of 38 ATP. 34 just from this one step alone. The most ATP by far that's made is in the electron transport chain. But the electron transport chain cannot take place if you didn't go through the Krebs cycle and get all those electron carriers and get the initial steps that went into the Krebs cycle. So that's all needed, but we just gain the most ATP from that last step. Now, I like this diagram because I can kind of somewhat draw it. My note, I think this is your book drawing. You know, if you look at your ebook and you're like this. I saw this when this new edition came out. I'm like, that is the most horrific thing ever, and there's no way I could draw it. And there's just so much going on that you just don't need to know. But you know, this thing is like that thing, and I was like, oh, whatever. So I was like, there you go, but I like this easier. Now, the electron transport itself, again, it can happen aerobically. It's where you're going to get most of your ATP, or the highest amount of ATP, but it can happen anaerobically. Again, I love Big Bang Theory. So many memes show up in my PowerPoints. So aerobic respiration, you just had that final electron acceptor that picks up the last of the electrons after they're done doing their job, is going to be oxygen. That's where we get our water. But it can happen anaerobically as well. It just means something other than oxygen is going to pick up the final electron acceptor. Some inorganic molecule, just not oxygen. The electron transport chain can also happen in fermentation as well, which we're going to get into fermentation. The whole, you know, going through and pumping hydrogens across can happen in fermentation. The only note, the final electron acceptor in fermentation is going to be an organic molecule, meaning a carbon-based molecule. And sugar is a carbon, you know, C6, H12O6. So it could be some type of sugar, which a lot of times it is. That leads us into our fermentation. And I'm going to come back. My note, you know, this big molecule, that glycolysis step that gave us the 2-pyruvic acid at the bottom of glycolysis, those pyruvic acid could make acetyl-CoA, could go into Krebs cycle, could go into the electron transport chain if it's happening in cellular respiration. But those 2-pyruvic acid could also go into the step of fermentation. So there's your pyruvic acid, and it can get further broken down. My only note is that fermentation does require NADH. I shouldn't say that. It makes NADH to feed into this. So to make NADH in our glycolysis step, breaking down glucose, NADH started out as NAD+. Can't make NADH, you know, an empty taxi cab, it hasn't picked up electrons yet. You can't make any NADH without first having NAD, or NAD+. Fermentation gives us some NAD+, which then can be used in cellular respiration, so they do go back and forth with each other. Fermentation is just a partial oxidation of sugar, which means we don't completely break down whatever sugar we put in. We would take glucose, break it down to pyruvic acid, and that pyruvic acid will get further broken down. But we don't lose all 6 of those carbons as CO2. It is not a full breakdown of sugar. Which means you're still going to have an organic molecule, because you haven't lost all the carbon to carbon dioxide. You're still going to have an organic molecule that can pick up the final electron acceptors. Some of our end products is we'll make some CO2. We do make some. And we're going to make some type of organic product. I'm going to come back. So if we take pyruvic acid, there are multiple ways we can break down pyruvic acid into multiple different types of end results. This is just showing 2 examples. We can take pyruvic acid and we can break it down and we can get lactic acid. And lactic acid is going to give us that NAD+ that can go right back into glycolysis. Pyruvic acid and break it down and eventually make ethanol. Why is this good? What is ethanol? Alcohol, people. It's alcohol. You guys all under 21? It can make alcohol. Again, pyruvic acids, breaking down sugar, we can make alcohol. That's how we make alcohol. And it also, in the whole process of breaking down, makes us some NAD+ that can eventually go back into making NADH in the glycolysis step. Different organisms take pyruvic acid and make different things. Our cells, when they take pyruvic acid, they will make lactic acid. Any guess how you know you've made some lactic acid? Because your muscles do this. Yeah, like they hurt so bad the next day after work. You're like, oh my God, I'm so sore, everything hurts. Lactic acid means your cells did not have enough oxygen. Your cells in that moment did not have enough oxygen to take the sugar and go through this whole step to give you energy. But you still need the energy to do whatever it is that you needed to do at the time. So instead, your cells took that sugar that was in your body and went through the fermentation step, which still gives you 2 ATP in glycolysis. So you still had the energy to do whatever you needed to do. The only problem is they went through this process and they made lactic acid. Now you're sore the next day. Now, our cells make lactic acid. Other organisms can make ethanol. What would happen if our cells made ethanol when you worked out? You'd get drunk when you worked out. It would be really intriguing. I don't know, maybe it'd be busier at the gyms. But our cells do not. Our cells only make lactic acid from pyruvic acid, but other organisms make ethanol. This is an example of different organisms and what they can make from that pyruvic acid. So there's a bacteria called Propionibacterium that takes pyruvic acid, and it makes carbon dioxide, and it makes an acid called propionic. This is what gives the flavor of Swiss cheese. The holes in Swiss cheese is because of the carbon dioxide that's given off. That was just little air holes. And that sharp flavor, that dry flavor of the Swiss cheese is from the acid. So only this particular bacteria is used to make Swiss cheese. Aspergillus, Lactobacillus, and Streptococcus, all 3 different types of bacteria and or yeast, or fungus, are able to take pyruvic acid and they can make cheddar cheese. So sharp cheddar is because of the strong acid flavor from the bacteria. Yogurt and soy sauce, it's all because of the lactic acid. So we make lactic acid, so do other bacteria. That gives a sharp flavor to the foods. Saccharomyces is a fungus that's able to take pyruvic acid and make our alcohol, our beer and our wine, any other kind of alcohol. Clostridium, same genus of bacteria where we find C. diff, but different bacteria. The same genus can take pyruvic acid and make acetone. It's your nail polish remover, or isopropanol alcohol, your rubbing alcohol. Now, although the word has the word alcohol in it, can you drink it? Like ethanol? This is toxic to your liver. It will destroy your liver and it will kill you. Don't drink rubbing alcohol, even though it has the word alcohol in it. Alcohol just means it ends, the chemical formula ends in OH. That's it. Different organisms make different things from that pyruvic acid. Now, because I don't have too much more, most of what we just got done talking about was catabolism, breaking down specifically sugar. However, there are some anabolic pathways as well. So catabolic pathways break things down. So what do anabolic pathways do? Or make things. So catabolic pathways, which we talked specifically about breaking down of glucose, break things down. Anabolic pathways build things up. But where does the energy come from? Because if you're going to build anything, if you're going to build a building, where does the energy come from? Well, it comes from catabolic reactions. We broke down glucose. We made lots of ATP. That ATP can then be used to build other things. Most of the time, these reactions, not all the time, but most of the time, these reactions are reversible. We can build things up, but then we can break those things right back down. So a reaction that can go both ways, because they don't all, reaction that can go both ways is called amphibolic. So this is just showing a single molecule called A and a single molecule called B. We can put them together into a larger molecule called AB. But we can also take that large molecule, AB, and we can break it down into its individual parts, A and B. So we can build things up and we can break them down. Although the reaction goes both ways, different enzymes are required for different things. One enzyme is needed to put them together, a completely different enzyme is needed to break them down. Integration and regulation of metabolic functions. It's so wordy. my whole kind of summary of this slide is these are the ways that your cells try to be as efficient, aka lazy as possible. So how cells try to be lazy. One, cells are only going to make enzymes because we don't always have enzymes around. Cells are only going to make the enzyme if the substrate is around. My example from before, the cell is not going to make fructose 1,6-biphosphate aldolase if the fructose molecule is not there. We're not going to make the enzyme if the substrate's not there. It's like, why do it? That's just a lot of extra work. One, cells catabolize, break down, the more energy efficient choice. So if there's two things that can get broken down, two different types of sugars that can get broken down, the cells are going to pick the more energy efficient one. We get more bang for our buck, more ATP from breaking down glucose than from other sugar molecules. So the cells are always going to take the more energy efficient choice. Cells are going to synthesize or make metabolites as they need them and then stop making them when there's enough. It's like, let's make the things we need and then let's just stop when there's already enough of it. And that feedback inhibition, where your final product becomes an inhibitor, also helps slow things down. So when you start making your product, that product, it goes back and stops the making of it. So when we make enough of it, we stop making it when it's abundant. It's like if we had enough of those little green ovals, it becomes an inhibitor, so we don't make any more green ovals. We got enough. Again, it's how our cells try to be efficient, aka lazy. Let's only make things when we need to make them, and then let's stop when we've already made them. And then I threw this in there because it came from your book. Everything that's got a purple arrow is a catabolic pathway. Everything that's got a green arrow is an anabolic pathway. The only thing I just wanted to note on it, because it's very complicated, is just note that as we make things, even just like pyruvic acid or any of the things that are needed to get to making pyruvic acid, that can be used as part of an anabolic process and a catabolic process. So it was used, we were breaking things down, but pyruvic acid could get mixed with something else and it's used to build something right back up. So a lot of the intermediate things that are made in the breaking down of glucose can be used as building blocks as well. So lots of breaking things down and building things up in our cells.